thank you everyone to come to the tennis talk and support Mizuki in person. So Mizuki comes from Bucknell University in the US, and he works on the largest amphibian in the world. So the Chinese one, Japanese one, and the Helvetia in the US. So he just jumped on a plane last night and took an 8 hours flight, arriving 7 a.m. And then drive the car, rent a car, coming down to YouTube. Luckily, found a parking space and figure out the way how to pay for the parking space. Luckily, music is here. So thanks so much.
Well, good to see you, everyone. As Becky introduced, I just got here this morning and drove a couple hours, hour and a half, and got enough coffee. But I'm a little tired, but I think I can do this. So just introducing myself. I'm from Japan. I don't know how much you know about Japan, but I was born in Northern Island of Hokkaido, right here. Oh, interesting. And then my parents are from northern part of Japan again here. They're school teachers and they couldn't find jobs in northern part of Japan where economy is not doing great. So they moved to Tokyo. So I grew up in Tokyo, but I have a strong sense of belonging to northern part of Japan because that's where my parents are from. And during sabbatical, I stayed in Kyoto at the university in Kyoto and taught there. And then I did research at Hanzaki Research Institute. And Hanzaki is a local Japanese name for giant salamander. And I also do research in Gifu area now too. So this is sort of places I am familiar with. Right, so I did my undergrad at Tsukuba University, Tokyo, did master's in forestry in Tokyo University and moved to America when I was 27 without speaking English that much. And over lunch, we talked about the challenge of doing science in the second language, but I definitely been through that and I am still struggling with English. So I did another master's in biology and then PhD in biology from University of Memphis. And then now I am associate professor at Bucknell University, and no one knows that Bucknell University, one of the liberal arts colleges, smaller college, I think 3,600 students on campus, and mostly undergrad students. And just for your information, because many of you are interested in teaching job, I don't know if liberal arts colleges exist in Australia, but it's a primary teaching university. So we tend to spend more time teaching than doing research. That's what they expect us to do. So I teach about 5 courses throughout the year, which translate to 6 to 9 hours per week in classroom setting. And then I have 25 to 30 advisors, which means I have to make sure those 30 students to take courses that they have to take to graduate. And in my research lab, I have about 5 to 10 undergrad students. Again, the university is primarily undergrad students. We have a smaller master's program. So I have 1 master's student in my lab, but mostly working with undergrad students. And I take my students to Japan to work on giant salamander research over the summer. And then in addition to giant salamander research, I do work on local species such as spotted salamanders and wood frog. All right, so that's enough introduction. I don't know how much you guys know about salamanders, but here's the phylogenetic tree of salamanders. And there are 10 families. And as you can see, this phylogenetic tree is color-coded based on the mode of fertilizations. So these 3 families do external fertilization just like fish do. Lay eggs and then males spread sperm over eggs and fertilization happens externally to their body. And then internal fertilization evolved once in the evolutionary time of salamanders, which has spread over the newer families. And as you can see, so Cryptobranchid is a giant salamander family. And up until 5 years ago, there were three species within the family. Chinese giant salamander, which Becky works on, and Japanese giant and American giant salamander helping. However, over the past 5 years, there are a few influential papers came out of Chinese scientists claiming that there are more species than one Chinese giant salamander in China, which is not surprising given how big the country is. And it turned out at least 5 distinct lineages should have existed in China. But this data is really interesting because this is the haplotype distribution of the wild specimens. And as you can see, color coding is really clearly divided into different lineages. But if you look at the farm individuals, in China, people eat salamanders. So people catch them, put them together from different areas and create hybrids. And to make things worse, they release those hybrids without knowing they're hybrids, into the river. So-called genetic pollution might be occurring in the wild habitat as well. In addition to this article, a more recent article found purebred new species of Chinese giant salamander, jansensis, and they collected 20-some specimens from the wild habitat and belong to the same clade, which established new clade in addition to the tree that I just showed you based on that previous paper. And according to the analysis, there should be 7 distinct lineages. So far, they named 2 additional Chinese giant salamanders. So the current species designation is there are 5 giant salamander species within family Cryptobranch. 2 new Chinese giant salamanders, South China giant and Jiangxi giant salamanders, and again Japanese giant and Help. So this is where we are in terms of classification of giant salamanders. And talking about my species, Japanese giant, it is listed as vulnerable under IUCN, which has been updated from near threatened last December. And I was part of that assessment team, and I definitely made a recommendation to move that classification to vulnerable. And then it's also listed under CITES Appendix 1, which makes my research really challenging because I can't even take DNA out of the country and bring it back to my lab to do analysis. And we don't have CITES designated institutions in Japan, which is crazy. So I can't really, there's no way for me to bring samples at this point. So I have to do analysis in Japan, or I have to ask people to do the analysis. And there are 2 Japanese laws that protect the species. One is by Ministry of Environment. This doesn't really have any power to protect the species actually, but the other one designated by Agency of Cultural Affairs, which is really interesting because they are the cultural people, but they see the species value as a cultural importance. Therefore, they put the highest protection as special natural monument. And this law is really strict. Because of this law, we have to get the permit from Cultural Affairs Department, which can take a lot of time, and often those people don't understand ecology or nature. So let's say we do microchipping individual, and they don't like that because they think that's bad for the animal, bad for the study. So it's hard to work with the people who are in the cultural division who don't understand ecology and nature. So that's what we have to deal with. And there are few major pillars explaining the declining trend of the species. Dams and water management, concrete banking is the biggest one. It's really depressing to go around and look at the habitat that are not good at all. And also, I'm going to talk about this, but hybridization with Chinese giant salamanders is happening, which is unfortunate because both of them are threatened species. But when they become hybrids, suddenly they become sort of not useful. So we need to take them away and then we have to kill them. And also we don't have enough people studying giant salamanders. Over the lunch, we talked about how getting a job is becoming really difficult, especially when you do pure ecology. Somehow, universities don't have those jobs. Therefore, we don't have enough scientists, ecologists who are studying basic ecology of this species. Here's a typical stream where giant salamanders exist, and it doesn't look anything special. You can see concrete banking, you can see some dam, but if you know the spot, there are a lot of them still existing in those limited locations. Well, by the way, so here's me. Well, this is me right there. You can see my butt. And then, in 10 minutes, 20 minutes, if you're lucky, we can find them. They're nocturnal species, so often day survey can be difficult. But if you know the sites, it's not difficult to find adults during that day survey. And this picture shows nesting site, which is really difficult to find. And here's the nest. And then underwater, if you are lucky, there's a bed master during the breeding season protecting the nesting site. And this is the actual bed master of the nesting site. And we are fortunate enough to find a nesting site where the entrance is close enough for us to videotape the behavior inside of the nesting site. And I can show you what's going to happen when we try to film this guy. Of course, and they have very sharp teeth. So if you stick your hands, if you get bit by these guys, you're going to be bleeding. And sometimes they do death roll. A lot of them don't have fingers. A lot of these guys don't have fingers because they bite each other, which I'm going to talk about. So you don't want to do that, especially during the breeding season. Males are ready to fight against any breeders. I wonder if you can spot a giant salamander. Yeah, there's one right there. And as you can see, there's a whitish sand in front of the nesting site. And that's one most supported evidence of nesting site, because often we don't see animals, right? But you see the white sand in front of the nesting site, and that indicates nesting site is close by. And then we wonder what's going on, why there's no sand. And people speculated a lot of different things, but we wanted to see what is happening within the nesting site. And this happens before spawning, before mating happens. So we collaborated with Asta Zoological Park because they have an artificial nesting site, and we put that cameras on, and this is what we saw. The male spends a lot of time cleaning the nest, which is amazing, kicking the sand, and you can see all kinds of debris is floating, and eventually this will be washed away from the nesting site. And interestingly, well, we spend my students spend a lot of time analyzing video clips. And then we found that the proportion of the time that they spend cleaning nest has increased up to spawning. And once spawning happened, the cleaning behavior stopped. And I wonder if you can think the reason for the cleaning behavior. Why do they clean? They clean much for the same reason we clean the house, which is to prevent infectious disease. If you have, dead animal carcasses or feces or whatever that organic materials that introduce water mold infection. And water mold infection is one of the biggest threats to eggs. Therefore, we think it's important for the den master to clean the nesting site so that when female comes, the sand is clean enough to have eggs without too much water mold infection. Here's a cartoon I created, and here's a den master DM main channel, and there's a nesting site. And breeding season happens late August and early September. When male comes, he gets really mad. And then of course fighting happens. And sometimes fighting gets to the level where death happens. And this small male, unfortunately, got killed by, our guess is by one of the den masters. And as you can tell, the throat is completely chewed through. This doesn't happen that often, but I guess this was, this individual was really unfortunate that they must have got that right weakest point and then their death row. Which made us wonder, like how much injury that they are suffering from. And because when we do research, like I said, we see a lot of missing fingers and limbs and bite marks and chips on the tail, but no one really look at injury rate. So we decided to work on the injury project last year. And it turned out there are not many amphibian studies looking at the injuries because I guess they are so small and they tend to die pretty quickly. But these guys can live probably more than 100 years. We don't know how long these guys can live because they live longer than us. So some of us started doing microchipping 40 years ago, and many of the individuals are still surviving. And the person who started that monitoring died a couple of years ago. So we need to have multiple generation of knowledge to know how long these guys can live. Yeah, another good thing about the giant salamanders is, when you see injuries, you wonder if that is because of predators, which is the case for many other amphibians. But these guys are the top predators on the river, and there are no other animals that can chew off the fingers and limbs, but than these guys. So we decided to digitize handwritten files stored at the Research Institute, which was a lot of work because, as you can see, there are so many volunteers involved in this monitoring. Some of them are better at writing and more precise than others, and it took us really long time. And there are some chicken written, languages that we couldn't decipher. But luckily, one of the students in my lab, Luna Hosley, whose mom is Japanese, was able to read language. So she really did a majority of work translating Japanese written file into English, and then Bridget helped putting the data together. And as you can see, entirely missing limb, some of the fingers are missing, again, entire limb is missing, some of the tails are missing, and those are the common injuries. So overall, we analyzed 684 capture records, consisting of 301 individuals. And then we found about 100, well, not about, precisely 100 are injured, which is 33% of the injury rate, which is pretty big. 1 out of 3 individuals that we capture tend to have injured. And then our prediction was small individuals tend to be more injured because they are the weaker males, weaker individuals when flooding happens. So that was one of the predictions. And then here's the data plotted against the efficacy. And as you can see, there's not the predicted trend. In fact, there's the opposite trend. Injury tends to be distributed toward a larger individual side. And when we run the stats, I know that figure doesn't look, significant, but when we run logistic regression, it was actually significant, which means body size has a positive impact on injury frequency. And then, this is of course different from our prediction, and we've thought about why that is the case. And I guess the most reasonable explanation is that they tend to live longer. They can regenerate fingers, but it takes really long time. And I don't know how many times they can regenerate fingers. Once they lose their limbs, I don't think they can regenerate. So the longer they live, they tend to have more chances to get injured, and that's our explanation. And then some of the small individuals are not really sexually active, which means they don't really fight yet. So that makes one of 22. And also, the fact larger individuals tend to be more injured means it's not because of the predators, because larger ones don't have predators, but because of the male competition or intraspecific competition causing injury in these species. And then another prediction we made was that males are more injury prone, sure, because male compete against each other to get the females. And here's the distribution of the sex. Unfortunately, a lot of individuals, we don't know sex. It's hard to tell sex, and I can show some slides about the sex in females and males. And it doesn't look like much trend, but if you look at the numbers, 142 non-injured unknown and 61 injured. In males, about equal proportion of not injured and injured individuals. In females, proportion is matching with unknown sex individuals. And when we ran chi-square test, it was significant. So male tends to be injury prone compared to unknown or female proportion. Like I said, sexing can be really challenging. And the only time we can confidently sex these individuals is during the breeding season. And as you can see, the cloaca of the male is really swelling during the breeding season. And when we see this, we are very confident this is male. Compared to the female during the breeding season, you don't really see swelling cloaca. And then also the stomach is bloated, which indicate female is yolking their eggs and ready to spawn. But there are some males that are not really ready to mate, and they look like female, and those are the tricky ones. So it gets like things are not this clear at all. The gray zone, and I can't really sex. So we decided to work on a DNA-based, genetic-based sex determination, by using the primers actually developed for Chinese giant salamander sexing. And it worked for salamanders, the American giant salamanders, and it actually worked for Japanese giant salamanders too. As you can see, we did a multiplexing PCR. This is a control and DNA, and this is a female sex-linked marker. And all the females turn out to be positive, males are all negative. So it's promising to use the primer set that are developed for Chinese giant salamanders, or both salamanders and Japanese giant. So we want to do this to unknown individuals living in that river. So yeah, again, prediction was smaller males are more injury prone. And then when we put both total length and sex in the stats model, total length was still significant, but the sex was not significant. So there's no really effective sex on frequencies of injury, which is different from chi-square testing. And we are wondering what's going on, and of course in different stats. But more than that, that maybe males tend to be larger. Therefore, the effect of sex disappeared when we incorporated total lengths as the variable. So we look at the males and female total lengths. The male tends to be a little larger, but it was close, but it was not significant. Yeah, but still, I think the body size difference between males and females explain less variation explained by the sex. And you might be wondering why I use total length rather than body mass. And of course, there is a strong correlation between total length and body mass, and I made both models, and based on the AIC, using total length was better than body mass. That's why we picked total length, which is a little surprising because body mass tends to be better measurement to me, because salamanders doesn't get straight up often, right? You have to wait until they get relaxed and they get really elongated totally, which doesn't happen often. So depending on who measure the salamanders, measurement gets a little little different. Injury types, you might be interested in this, and this is the types of injuries. We found that those are the numbers. Some of them have head injuries and back and tail, limbs, left and right, and you can sort of see the trend. It was really interesting to see they tend to get more injured on the right side. And also they tend to get injured on the hind limbs. Which we really did not expect it, but based on the data, these guys are right-handed. They tend to turn this way and then somehow get beat by the opponents. And sort of my simulation is fighting happens and they tend to turn left to face the right side of the body to the opponent. And then as they escape, they get beat by hand limbs. And that's how I sort of interpreted injury pattern. And I talked to some other field giant salamander people, and they don't agree with me. So I still have to look into this. But it was significant, and some of you might know, but handedness is actually pretty common across all the different taxa of vertebrates. And handedness has been studied for monkey, primates, and then also fish. And some of the fish tend to attack the prey from the right side and left side, and it depends on the population. It's a frequency dependent selection, because fish that are attacked from the one side learn to escape. But if there are other individuals that attack from the other side, they tend to succeed in capturing fish. So wonder if the same thing is happening to these guys. So finding to date, again, about 30% individuals are injured, and larger individuals tend to be injured, and males tend to be injured, although it was not completely clear. And then right side feet, toes tend to be missing, and hind feet, toes tend to be missing. And I still need to do more analysis and looking at the relationship between injury and BMI, and then also injury scores. So my data analysis is based on presence absence of injury, but I didn't really count how many missing toes. But we have that data too. So we want to use the scores in addition to presence absence of injury. Okay, so get back to this cartoon. Fighting happened, right? And then that's why I started talking about injuries. But of course, when female comes, their muscle is really happy. And then the female in and spawning happens. But interesting thing is, when spawning happens, the sneakers are going in. And they started mating together, all together. And then master, at this point, doesn't get upset. He just spray spawn, keep doing, no fighting. And I guess other sneakers know that's what that master does. So often one female versus like 3, 4, often sometimes like 10 males get together and spawning happens. And we don't, you wonder parentage of eggs, right? And we wonder too, but no one knows. We assume they must explains majority of the offspring, but you never know. And after spawning happens, well, okay, so I do eDNA stuff, and this is interesting because spawning happened during the breeding season, fighting happens, and baseline metabolism increases, which let us predict eDNA in the water should be higher during the breeding season because a lot of things going on. And this is the study we did based on Hellbender eDNA. So as we predicted, during the breeding season, eDNA concentration really increased sharply. And we also predicted eDNA during the nighttime is higher than daytime because they're nocturnal, which was not supported by the data. I guess they don't really move even during that nighttime. They're nocturnal, but they don't move that much. And then we found the same trend in Japanese giant salamanders, which has not been published yet. Right, so everyone, after spawning, everyone leaves except for their master, and stays, their master stays with the eggs. And we wanted to see what he is doing to protect eggs. And of course, he protects eggs by biting and attacking intruders. But their master does more than protecting eggs. And this was the most common parental care behaviors that we observed. Tail and if you study fish male do the same thing to their eggs. And this is the behavior to send oxygenated water to the eggs for the normal development. And we were fortunate to find two nests, one in natural environment, the other one in the artificial nesting site that we installed. And as you can see the numbers, this is the proportion of the time that they spend doing tail fanning. So 40% of the time we recorded is male spend while male did tail fanning versus in natural stream only 1 to 2%. And of course, the sample size is so small, but again, finding nesting sites is one challenge. Another challenge is to videotape these guys in the nesting site. This was sort of convincing to us that tail fanning is related to sending oxygenated water, because if you look at the difference between artificial nesting site and natural nesting site, within the nest, there is much less dissolved oxygen in the water. So the male probably sends that level of DO and then try to increase the oxygen in the nesting site. In fact, this male actually gave up on the nest during our study and he left. He disappeared completely and most of the eggs went back. And our sort of interpretation is the nesting site. The nest was not good enough for him to stay and then take care of eggs. Here's another interesting video clip of parental care behavior. This is a little long video clip with the wonderful footage. We collaborated with a wildlife photographer, Mr. Fukuda, and he sat in the river 6-7 hours a day and then took all the video clips. And we, as ecologists, we are not that good. We can't stay in the river that long, but he did. And he captured this video clip. Wonderful, really valuable. And I'll let you see the video clip and think what this individual is doing. And as you can see, eggs are connected through strings, and there are 2 strings originally, and for some of them separately broken. You can see some of the embryos are already developing and moving inside. It takes a little longer time, but it's interesting action will happen after this. And he's still doing something. Oh, by the way, you can see some of the eggs are whitish, right? And those eggs are dead eggs, either not fertilized or not developed normally. Okay. All right. Isn't this amazing? This is so exciting to watch. Well, so what happened? He actually ate the egg. He was like holding one egg, and finally he got the thing out of the string. And we repeatedly observed he was eating eggs. And we tried to identify like what are those eggs. And those eggs are that whitish eggs. So he was actually sniffing around and finding dead eggs and trying to remove those dead eggs out of the clutches because because those dead eggs attract water mold infection. And once they get the water mold infection, that can spread over healthy eggs. So he must be cleaning the nest. But Hellbender researchers, they found the same behavior, but also Hellbender males eat healthy eggs too. So depending on the condition, depending on, we don't fully understand what is affecting their behavior and decision making of eating dead eggs versus eating healthy eggs, but they do this behavior, which is fascinating. Very cute. Photo. I love this photo, like almost smiling. And these are toes. Their toes are so cute. You don't think these guys are cute, but if you see them, they're so cute. I don't know if you can see babies. Babies are here. There are babies there. Babies here. This is the dad protecting babies. So even after hatching, male stays and baby stays with male, and then male provide same parental care, which means eating dead larvae. And occasionally, this is again, photographers say there forever into this amazing shot. He ate a live larva, and then he actually sort of tasted the larva inside of the mouth and spit that out. And this is, according to the photographer, when the spitting out happened. So he can tell in the mouth the one that he ate was actually alive again. What a wonderful body. It's amazing. So spawning happens the late August, beginning of September, and hatching happens about a month later, and then dispersal happens in the following spring. So male stays with their eggs and larvae up to 6 to 7, and then finally these guys are ready to disperse. Okay, so that's the sort of parental care project that I've been working on for the past 10 years. And then I'm going to talk about the newer research. I know I don't have that much time, but let me talk about one eDNA research. So, like I said, we saw larvae with power, but that's the sort of only time we get to see a lot of larvae. Once they disperse, it's very difficult to find them. We actually find more adults than larvae, which is not right because we need to find more babies than adults. Right? But that's not the case with these guys. So we wanted to see where those larvae are. And we predicted that larvae might be using, or small individuals might be using very small tributaries. And here's the main stream right next to the Research Institute where I do research. And a lot of research has been done on the main stem, but not the tributaries, because tributaries are so small. And I can show you. And so we surveyed 3 tributaries, A, B, C. And this is one of the tributaries. It's like surrounded by forest, middle of nowhere. And then stream width is very small, like 1 meter, 2 meters. And given their body size, we wouldn't think they are living here. And if they're living here, we should be able to find them. And we collected water samples from different tributaries, again, really small tributaries, and put them in a cooler, doing filtration at the kitchen of someone's house. But then we did not find any salamanders here in the water sample. So, I thought, okay, there shouldn't be there. But when we did analyze eDNA, by the way, eDNA is okay for me to train, but I talked to Fish and Wildlife, I talked to the American, the Japanese government, they said it's okay to bring eDNA because we are not harming any product line, it's just more science. And as you can look at that there, most of the sites are positive, and the X indicate negative sites, and only less than 10 sites are negative, which means we found eDNA from almost all of them. And you can see there are 2 interesting peaks there, and we are wondering what's going on at that site. So based on this data, we decided to go back to those sites to see what happened, like what is the source of high eDNA concentration. And this was one of them. And it doesn't look very good at all. You see the concrete, 3 faces concrete, the bottom and 2 sides. And Brianna, my student, is taking water sample, and that's the water sample. We got really high EDNA concentration. So I'm like, okay, EDNA is not really reliable, right? But if you look at the picture, there's a little bushy area. So we decided to go back to the bushy area, and it actually looked good. And you can't really tell in this picture, but I saw the whitish sign right there. So which indicates nesting site. So we came back with that camera. So the camera is attached to the stick, and Sunil and my collaborator is looking at that inside of the nest, and we found eggs there. So that was actually nesting site. That's why EDNA concentration was really high, because the larvae tend to be hiding in this little space. We actually found the den mustard there too. So that was a good news. And then we wanted to see what's going on to the other peak. And this was the other peak, and it really didn't look like promising at all. But we found a little, mossy area, about 10 meters above the sampling collection site. And mossy area is a good indication of stable habitat, because Japan is very steep terrain, a lot of rain, and then this provided stable habitat. So we wanted to see if there are any larvae or juvenile hiding under these rocks, and we fled these rocks. You can see some of the rocks are already fled. About half of the rocks are fled, and we spent about 30 minutes. We didn't want to disturb that area, entire area, and we found one juvenile. And this guy is 38 centimeters, but still juvenile, not sexually active. And then we found the larva too. And again, finding larva in small individuals is difficult. And based on the EDNA concentration, we got to find the larva and the juvenile. So I think that's a habitat containing all of the individuals. That's why the EDNA concentration is higher. Okay, and then we did a nice survey, and we actually found quite a few salamanders. The red indicates salamanders. So EDNA, well, so this is a histogram of the individuals that we found. And the body size of these guys are much smaller than the ones we find in the main stem. So we know that the smaller, less competitive individuals tend to use small tributaries. And once they become larger, they might come back to the main stem, because they need to prey upon larger prey items. And that's how they use small tributaries and large main stem. So this is the last slide, but based on the EDNA survey, it was great, because even undergrad students can collect water samples and do analysis. They are not very good at doing field work, but they can do water collection and deep extraction and stuff like that. And then we found that these guys actually use small tributaries. So it's important to conserve not only the main stem, but also small tributaries. And we tend to ignore small tributaries, and we tend to make dust between main stem and small tributaries, which is really bad management. So that's one of the recommendations we talked about in the paper. Okay, I think I should stop here. I have more slides, but I know the seminar is ending in 5 minutes, and I should take some questions. So thanks, guys. Do you have any questions, please? Yep.
I also wanted to a comment to say that I work on sailfish, and they do have that left and right sided attack lateralization. Cool. And I'm also hoping to do stuff with sex markers. So it's interesting to know that they're doing that with the giant salamander.
Right.
Do you know if there's if with your video of the nests, it just sort of reminded me of the Murray cod project that I heard about. Is there like predation happening, I guess, still, like, is that
predation of eggs?
Yeah.
Predation really did not happen.
Mm-hmm.
The larger like fish and turtles started to get into the nesting site, but because of the den mustard, they can't really eat those eggs. So water mold infection, I mean that can be predation, because they are eating eggs. That might be the biggest predation that can happen to those eggs. Insects, aquatic insects, probably not, because eggs are pretty big. So again, we did not observe bigger predators eating this, but watermelon.
And also with the video stuff, do you think, I guess with the dens, is it more of a competition for females versus like a competition for space? Or
Yeah.
is there a lot more den habitat?
Very, very good question. So competition for spaces. There are limited number of suitable nesting sites. So males compete against each other to get to be den master. And then, of course, you know, if they occupy suitable dens, a lot of females come and lay eggs. So eventually that lead to competition for female, but competition initially happens for the species. Yes.
So multiple females visit one den master.
Yes, multiple, yeah, very good question too. So, right, so one female lays 300 to 500 eggs. And we often see more than 500, which indicate that multiple females are actually visiting the same den master. So the den master, I don't know if the den master is good or nesting site is good, but they tend to attract more females.
Awesome work. Obviously, having no use of salamanders on this continent makes me a bit jealous. I was actually just thinking about Caitlin's research. She works on crocodiles and their social stuff.
Yeah.
Yeah, I was thinking about your findings with the injuries.
Mhm.
And I know, Caitlin, you know a lot more about this than me, but the larger crocodiles will tend to have more injuries because they defend the better sort of social spaces. And
Yeah.
I wonder if you thought about similar social dynamics with the den masters being like a kind of behavioural trait. Maybe larger den masters are getting more injuries that way if something remains.
Yeah. Yeah, another good question. And as far as we know, these guys are not really social species. So we don't even know if they recognize their kins, which like some amphibians, they do recognize kins. And the ones that cannibalize each other, they tend to avoid eating their sisters and brothers. So it might happen to them, but again, we don't really have that DNA genetic data to identify kins. And also that leads to another interesting interpretation of communal nesting. So those sneaky males might be their brothers. If that's the case, it makes sense for them masters to invest that much time and take care of babies. But we don't know that kinship. Yeah.
You know what a lot of the females conspecifics do in the meantime whilst the males are protecting the nest?
Can you say that again?
What do the females do after they've laid the eggs and while they're protecting the nest?
Yeah.
Go and feed or hunt, that sort of thing?
So we think females might be actually mating with more than one male. Yeah, that's our guess. And again, we don't have any data tracing females and finding the evidence of multiple clutches dropped in the different nest sites. But it's very likely females are doing that.
Thank you.
Mhm.
Do you know how much time it takes for a male to make his den, and then if he dies, I'm presuming it's heavily sought after because you said it's limited size.
Right.
And can new ones form depending on the river changing or anything like that? I saw that a lot of it was channelized, but in more natural ones, are these things changing all the time and take a bit of effort for them?
Yeah, so they do modify structure of the den a little bit, but they can't really do too much. They get rid of small rocks and clean the sand, but mostly they have to depend natural structure being suitable for nesting site. And as you said, we have a lot of flooding, terrain is very steep, so the one flood can destroy nesting sites. Sometimes nesting sites stay multiple years, but they can disappear. So it's really dynamic relationship between male finding nesting sites and making a little space and then disappearing and appearing of nesting sites.
You don't know how long it takes them to make that little cavity or?
Yeah, well, so usually males or den master occupy nesting site at the beginning of August or mid-August. So dead mustard stays there up to month, 2 weeks to a month. So that has to be the period where when dead mustard is doing cleaning and making that nesting site. Yeah.
Does the same den master return to the same den the next year?
Yeah, they do, but because they don't eat during parental care, they tend to lose a lot of body weight. And again, we don't have a good enough data to show that, but our sort of observation is that after being den master a couple of years, he's too tired to be den master, so they retire from den master and become sleepy for a couple of years and gain energy. They come back to den master. So den master is not really permanent running, but I think they rotate depending on the body condition, body mass. It's a really costly activity.
Well, first off, this is just fascinating. It's super interesting stuff that you're finding out here about this really cryptic and I guess really not studied species.
Right.
You made a point at the beginning talking about how, you know, this is pure research. Like we're interested in these salamanders, we're learning about these salamanders because they're awesome.
Right.
But there's also, you know, obviously a clear applied component when you mentioned that the IUCN ranking has been uplisted to vulnerable.
Right.
So I wonder if you could speak to the experience of going for pure research or getting funding for pure research when there's such a push for an applied angle for our research.
Yeah, well, so when I get on tenure track position, I started working on these guys, knowing I have a little more job security. Once I get tenure, you know, I can invest more into basic research. Yeah, but you're right, though, a lot of people want to see applied research. But again, applied research is based on basic research, and we don't have basic research about these species. So what I'm trying to do is, in the discussion of this article, for example, this is really basic research, but we try to bring a lot of conservation applied aspect into the article. So we talked about the habitat fragmentation and movement of animals, which is not really what we studied, but that's implication from our research, which was important because even with that basic research, we can incorporate applied aspect into discussion, which is really useful for IUC. So this article was very useful when we did evaluation because no one else was doing research. That's really my struggle to do. Yeah, it's a struggle.
So I guess on that note, do you sort of draw the link then with the presence of salamanders with some of the eDNA work to like water or stream health and those sorts of things, waterway management? Have you made that connection?
Yeah, well, so so here's the dam, right? In that the dam, there are huge hydroelectric dams. I mean, in the picture, this is the size, it's huge, and this is the village, below the dams. If something happened to them, people will die. And I don't know how people are mentally dealing with this, but
Yeah.
we live in one of these houses when we do research. But anyway, so we're wondering if giant salamanders might be living upstream of big dams, right? Because they used to be connected to the main channels. So we did ENS stuff, collecting all the water samples, tributaries running into these dams. If they are still living, they've been fragmented for like 50 years, which is so sad for these guys. So we did DNA collection and filtration and all that, and we found 13 out of 23 positives. So they're still there in one of the dams, and 0 positives for this dam. In this dam, tributaries look terrible. A lot of concrete banking, and this park is called Eco Park. And the city made the Eco Park by making all the concrete banking. And of course, they can live. This is the best tributary running into the dam, but no evidence of health with Japanese giant salamanders. And we just collected 29 more samples from the last dam. So we do based on the eDNA, I can make some conservation recommendations. We need to do something about those guys, right?
Yeah, because you showed that they were quite picky, I guess, about how their den is, and the male had abandoned that artificial den.
Mhm.
So I guess the characteristics of the good den might also be characteristics of a healthy waterway, perhaps.
Yes,
Yeah.
yes, yeah, having good enough habitat for nesting site is definitely the most important thing.
Yeah.
Once again, these guys can live, probably more than 100 years, which means even without recruitment, we tend to see those large individuals. And when people see large individuals, we tend to think, they're doing okay. But that's not true for, those evaluated species. And when we can't find small individuals, that's a more concerning situation.
There's a bit of a trend in Australia, and I'm not sure in other countries as well, but to de-channelize creeks and to bring nature back into cities and suburbs and that, and with some pretty good success in just removing the concrete and letting plants just, live, and all birds come back and everything, and it enriches people's lives. I was just wondering if you've tried that in, try to be a proponent of that, which would obviously, I presume, help the salamanders there.
Yeah, that would be wonderful. Unfortunately, Japan is behind in terms of understanding of how those buffer zones protect not only biodiversity, but also the river and people's life, because that can be a natural flooding the zone, right? So that can absorb the water and then protect the downstream people. But we don't, people are not doing that at all. And conservation education, environmental education in my country has been poorly done. Kids are not learning anything about environment, not anything, but very little. Like when I talk to kids, little ones to high school students, they know very little about environment. So, and not only ecologists, but the common understanding of how nature works and what is the best decision making, both for human and the wildlife, needs to be shared among larger body of people who's making decisions about concrete banking and city development and stuff like that. But those people have no idea. And some of the very small proportion of ecologists know, hey, you can't really do this, but our voices are not really reaching to them at all. Really frustrating.
To go off of that topic a little bit, I was interested at the beginning when you mentioned that under the federal government, Japan was listed as a special natural monument.
That's right. Yep.
And I think that's awesome that there's a cultural recognition for the species.
Mm-hmm.
But does that translate into the culture? Like, are people aware of the species? Because I grew up in Missouri where we have hellbenders.
Oh, yeah.
And most people have no idea what the hell it is.
Okay.
So I'm curious, is this a known critter in the environment?
Yeah. So, right, compared to American situation, I would say almost all Japanese people know about the species.
Really?
It's a really well-known species. They know at least the name. But
Is that partially due to this
yes,
cultural minister?
yeah, it's right, the protection by the cultural agency makes the species really popular. And there are a lot of books about that protected species. And also there are so many TV shows about this species.
Really?
Yeah. So people know about this species, but again, they don't make connection between their life history and how the habitat should be protected. So there's one Japanese shrine worshiping giant salamander, which is crazy. There's a little shrine, right? And you would think the people in the village protect the species. They don't. They use the species to sell goods, which is very unfortunate.
Commercialize this.
Some of the well-intended people trying to do conservation work in that place, but again, the focus is on the economy and how they can use the species to make money, not to protect the species. And unfortunately, Japan is behind of that scale.
And China's falling behind. I think maybe 90% of people don't know about giant salamander, even the biggest one,
Yeah.
and they only know about the delicacy.
Mm.
So we're using Japan as an example. Can you think
Japan is doing a little better than China, yeah. It's still not great.
I guess, sorry. On that note, so you mentioned that it was quite difficult to take DNA out of the country, and I know
Right.
that's the case for a lot of other places.
Yep.
Do you need to then extract the DNA? Can you remove extracted DNA? Or like, what are some of the issues with, I guess, these protections
Yeah,
and
Right.
research? Because that's something I've encountered as well.
yeah,
with
simply taking the tissue sample,
Yeah,
because you're harming animals by taking the tissue sample.
yeah,
So either that is a piece of meat or a certain DNA that is based on harming animal. That's why we can take that. Anything.
even
Coming
if it's
from-
their toes
Yeah,
and they
toes,
lose
yeah.
their toes.
We can't.
Yeah, okay, because yeah, it's interesting to hear that and then to hear that their habitat isn't being protected, and surely that's a greater harm than taking a little sneaky toad, but anyway.
No, I
Yeah,
totally agree.
that's frustrating.
You know, during the pandemic, it has become even more strict.
Mhm.
And when I traveled during the pandemic, I guess, you know, Asian biologists traveling is a red flag. So I got stopped at the airport much more frequently than before. Oh. And because my green card has my job descriptions, if they scan, they know.
They're looking for you.
Yeah, they're like, my they scan you abolish. Right, so they check with my bags and I know, so I don't take that risk.
Yeah.
Yeah.
Because, as I say, with the microchipping, I was interested to hear that that was a thing that was happening. If you have like known age individuals, it might be interesting to look at like DNA methylation methods for aging. And so that you might be able to answer the question
Right,
somewhat of how old do these things really get? And
yeah.
yeah,
Yeah, I think to make at least a few universities certified
Mhm.
would be a good step. But even Tokyo University, Kyoto University, those are two leading universities in Japan, they're not certified. And I don't want to go through the process which I would need to have to go through hearing and money and all that kind of stuff on both ends. I get that.
yeah.
Yeah. Yeah, but yeah, it's a challenge.
Yes,
It's a
thank
good-
you.
good regulation.
Right, thank you everyone, the wonderful 1 hour, but you feel free to hang around to talk to Mizuki